Our next speaker is Raquel Furtado, coming from the Albert Einstein College of Medicine in the US. So Raquel, please go ahead and start your presentation.
All right, hello everyone. I want to first of all thank the organizers for inviting us to present our work today on characterizing the immune response against malaria in patients that live in endemic areas. And importantly, the identification of a population of memory CD4 T cells that we found to be clonally expanded and have interesting cytolytic signatures. So next slide, please. So as many of you may be aware of the various Plasmodium species that cause malarial disease, it's primarily falciparum malaria that is the deadliest and accounts for 90% of patient mortality. And in the human host, this infection can be very generally parsed into two phases. One is the asymptomatic liver stage of infection, and then the symptomatic blood stage of infection. Now, both in humans as well as in mice, we know that antibody responses are very important for control of this infection and for the response of this infection. And indeed, in humans, we find antibodies generated against various P. falciparum antigens expressed at various stages of the Plasmodium life cycle. We also know that in humans, passive transfer of antibodies from immune donors to infected individuals can actually reduce disease burden as well as severity of disease. However, in terms of cellular immune responses in humans, it's still unclear as to which populations of cells are important for responding to the infection and which of those populations of cells are important for conferring long-term protection. In mouse models of malaria, we know that B cell responses, cells that produce antibodies, as well as CD4 T cells are essential for protection. But it's important to characterize these populations of immune cells in humans as well, so that it can inform the development of a more efficacious malaria vaccine. The current vaccine that is licensed against malaria, while important, only confers limited protection. So on the next slide, I just want to summarize that our rationale and hypothesis was that if we look at individuals who live in malaria endemic areas, who we know historically through studies that have been done, and also work that I will show you here today, those individuals seem to develop clinical immunity against P. falciparum malaria, likely owing to repeated exposures to the parasite, having lived in those areas through their lifetime. And we postulated that perhaps these individuals with naturally acquired immunity possess some immune populations that are mounted against P. falciparum that are either distinct in their frequency or in their function compared to other individuals who might be having their first exposures to this parasite. So on the next slide, I'm going to introduce you to our cohort of patients that we studied. We did this in collaboration with the International Centers for Research Excellence, ICMR, in Malawi. And in a village in Malawi called Mafera, we enrolled patients who had symptoms of mild malaria. Each of those patients that were enrolled in the study were then longitudinally followed for 18 months. And through active and passive surveillance, we were able to capture each time these individuals had a malaria infection. And when they had a confirmed malaria infection, we collected blood from those patients, both during the time of infection and at 30 days later at convalescence. From those blood samples, we were able to harvest DNA, plasma, as well as peripheral blood mononuclear cells that were then sent to the lab for further analysis. So at the end, on the next slide now, I will show you the outcome of this study. At the end of the 18 months, we had 97 patients who were retained in the study. And what I'm showing you here in this plot is the number of malaria infections each individual patient had, and I've plotted this here by the age of the patient. When the study was set up, we ensured to enroll patients across all age groups from the ages of 1 through 50 in hopes of capturing adult patients who likely had developed naturally acquired immunity, as well as young patients who are probably having their first exposures. And what I hope you can take away from this plot here is that we were actually able to identify 3 groups of patients. In pink are young patients below the age of 13 who had a very high number of malaria episodes since the time of enrollment. They had 3 or greater episodes. And then we were also able to identify what we defined as protected patients in orange and green. Green being the adult patients above the age of 13 who had no malaria infection since their enrollment in the study. And then the patients highlighted in orange who were age matched to our susceptible patients and had either one or just no infections through the study. So on the next slide, what I'm showing you are the subsets of those three groups of patients that we analyzed in depth further. So from the plasma of these patients at the time of infection, we were able to characterize their antibody responses, which I'm not going to share with you today, except to say that in our adult protected patients, we did detect higher titers of PF-specific antibodies, as well as antibodies that had higher opsonic activity against PF merozoites in comparison to the plasma antibodies present in our susceptible patient group. And then what I will continue to describe to you today is the in-depth characterization we did of the peripheral blood mononuclear cells or the immune cells in these three patient groups. So on the next slide, I'm going to show you the data that we obtained from our mass cytometry analysis, which is the approach that we use to characterize as many immune cell populations as we could that were present in circulation during infection. We did a high-dimensional mass cytometry analysis that covered 32 lineage and functional immune cell markers. And what I'm showing you here is a summary of that data in a t-SNE plot, which is a reduction of those 32 parameters onto a 2D plane, clustered based on their cell expression profile. And what I hope you can appreciate from the top panel is that if we look at the clustering of these cell populations in our susceptible patients in pink versus our protected patients in green, what we found is essentially distinct clusters of cells present in our protected patients compared to our susceptible patients. And if we annotate those clusters of cells in the bottom panel here, we were actually able to identify innate immune cell lineages as well as adaptive immune cell lineages. So on the next slide, I'm going to highlight and summarize for you what those main immune cell populations were. Firstly, we saw among the innate immune cell populations a higher frequency of classical monocytes among our age-matched and adult protected patients compared to our susceptible patients at the time of infection. And as we know, classical monocytes have high phagocytic capacity and important for response to infection. Further, we also saw a higher frequency of memory CD4 T cells, as well as a higher frequency of memory B cells in the top right panel, as well as a higher frequency of switched B cells in the bottom right panel. It's possible that these higher frequencies of memory CD4 and B cell populations are due to the age of the patient. But despite that, we wanted to further characterize this population of memory CD4 T cells because we know that they're important for responses to infection, especially viral infections. And we know that CD4 T cell responses are also important to promote a robust B cell responses and the generation of antibodies. So on the next slide, in order to further study these memory CD4 populations that are present in these adult protected patients at the time of infection, we conducted a transcriptional analysis through single cell RNA sequencing. And this method allowed us to basically obtain the transcriptional profile of every single memory CD4 T cell in 3 adult patients that we analyzed. So what I'm showing you here is a pooled analysis of the transcriptional profile of the memory CD4 T cells taken from these adult protected patients. And what we find is essentially distinct clusters of memory T cell populations based on their transcriptional profile. So in the next animation, what we were able to see in the next slide, is essentially expression of different genes that allowed us to annotate these clusters of cells. And what we found is that there are subpopulations of memory CD4 T cells that have regulatory capacity. We have those that have Th2-like function as well as central memory characteristics. But importantly, what we found is a population of cells that have an effector-like phenotype. And these are the subsets that are highlighted in green, of which you can see on the right violin plots, the dark green subset that's labeled as Th1 has a very high expression of molecules that encode cytolytic effector molecules, such as the Granzyme family of molecules and granulysin, as well as the transcription factor ZEP2 that basically can program those cells to have a very effector characteristic. So on the next slide, what we also wanted to determine is the clonality of these T cell populations that we profiled sequentially. And the reason for this is because if there is a population of T cells that is responding to the infection, they essentially would proliferate and share their T cell receptor, have the same T cell receptor. And indeed, across the 3 patients, we were able to identify expanded T cell clones within this memory population. And interestingly, among all the 3 patients, these T cell clones fell within this cluster of Th1-like cells. And this is summarized in the pie chart, where a majority of expanded T cell clones fell within this green cluster of Th1 cells, and only a small population of them fell within the regulatory subset. So in the next slide, I want to show you that these expanded T cell clones made up about 0.5 to 1% of the total memory T cells in the 3 patients. And in the right plot here, we found that they express very high levels of transcripts encoding effector molecules, such as Granzyme family of molecules, as well as the ZEP2 transcription factor. On the next slide, what we also found was that if we focused on the cells that express these effector molecules, it's mainly the cells that express high levels of Granzyme B that capture a majority of these expanded T cell clones. And this is surprising to us because while it's not too surprising to find expanded T cell populations within a patient during infection, it was surprising to find that these expanded populations within the memory CD4 T cells had these cytolytic features. So on the next slide and the next animation as well, what we found is that these cells can actually be detected in the patients across all 3 groups, not only during malaria illness, but also during convalescence. And we were able to confirm this by flow cytometry. And on the next slide, what we found among these cells was that they express chemokine receptors. And in the next animation, that they also express inhibitory markers. And what I want you to take away from here, from this slide, is that when we compare the 3 groups of patients, the main finding we had was that the cells that express Granzyme B, that are memory CD4 T cells in our protected age-matched patients, actually express lower levels of inhibitory markers compared to our susceptible patients. And these inhibitory markers, which include LAG3 and PD-1, are often targets of immunotherapies to reactivate some of these populations and invigorate their effector functions in T cells. So I'm going to skip the next slide for the interest of time, except to say that these populations can be reactivated ex vivo. And I hope that you can take away from my presentation that from our clinical study into our high-dimensional flow cytometry analysis and then our single-cell sequencing analysis, we were able to identify that during malaria infection, there is a clonal expansion of memory CD4 T cells that have cytolytic features, express high levels of Granzyme B molecules and the ZEP2 transcription factors. And on the next slide, we hope to show you that this data could then maybe lead to developing a hypothesis that these cells might be involved in direct killing of P. falciparum infected cells. We know that these memory CD4 T cells are likely responding to MHC class II expressing cells, and it's possible that they're probably responding to cells in the liver that express MHC class II. Our hypothesis is probably infected hepatocytes or maybe Kupffer cells that are presenting parasite antigens. So on the final slide, I want to first of all thank you for your time and thank the patients and families that participated in our study, the collaborators at JHMRI Malawi, my postdoctoral mentors Gregor Lobo and Johanna Daly, as well as Fabian Delahaye and Ryong Kim, who conducted the single-cell RNA sequencing analysis as well as the clinical data analysis. And I want to thank you for your time, and I'm happy to take any questions or connect with you in the future.
Great. Thank you so much, Raquel. It was great to see some work done from the Malawi ISOMER. We work just next door in Zambia, so it's always nice to hear. So there's quite a few questions. We'll have time for a couple. Do you, just in general, the patients, do you know, were you able to tell if any of the subsequent malaria episodes were reinfection or were any of them recrudescence?
That's a great question. So the way in which we try to ensure that is we only accounted for infections that are about a month apart. We also have DNA from those patients, so we could genotype the actual parasites. We haven't done that in our lab, but other collaborators within the isomer team are looking into that. But the way in which we try to sort of parse out that they're not reinfections is that we made sure that they were at least 1 month apart. It's possible that we are catching some that are reinfections, but we hope that by keeping that separation between one infection to the next, we're hopefully capturing just new infections.
Okay. And then there's quite a few questions about either concurrent infections or reactions to other types of infectious diseases. So do you know approximately what proportion of the memory T cells were reactive to Plasmodium as opposed to other infections? And did you look at any patients that were co-infected with any other diseases?
So first of all, just to answer the second question, in terms of co-infections, the main thing for the study was that the patients were screened for HIV, so we know that they're HIV negative. But in terms of having any other co-infections, we actually do not have that information. And to get to your first question about specificity of these cells, indeed, we are only looking at clonal expansion of these cells at the time of a confirmed malaria infection. And we do not have any direct evidence to show that those cells are responding directly to malaria, except for some preliminary ex vivo studies we've done where we have re-stimulated these cells ex vivo taken from these patients at convalescence, and we've incubated them with parasite antigens, parasite lysates taken from infected red blood cells. And we found that these cells do become reactivated, suggesting that they can recognize PF antigens. We don't have direct evidence. We would have to develop MHC tetramers and would have to select our favorite malaria antigen in order to detect that specifically. That is sort of out of the scope of this study, but we're hoping that future work could address that.
Okay. Maybe just one more quick question. Did you happen to look at the parasitemia levels of any of the patients and if there was any difference across the parasite counts?
Yes, we definitely have that data. And certainly the young susceptible patient group had much higher parasitemia levels, significantly higher parasitemia levels compared to our adult patients. So the adult patients at the time of infection, the cutoff was 2,500 parasites per microliter for confirmed malaria infection, but our protected patients certainly had a lower burden of infection.